Welcome to 1234 Media Bio. Please don't forget to hit the thumbs up button at the end of the video if you really like it. You can also comment down below for any kind of feedback or suggestion regarding this video, and yes, please do share our video with friends and family. You are like gold, so finally please subscribe to our channel if you wanna do some wonder for our beautiful family, and also click the bell icon so that you may never miss an update from us.
This is part 2 of the cell division series. Yeah, of course, if you really want to learn properly. So in the previous video we saw that there are 3 types of cell divisions. Mitosis, which occurs only in the somatic cells. Meiosis, which occurs only in the reproductive cells. And binary fission is what we will study in part 3 of this series. This is part 2 and we will study about meiosis. Meiosis is a process of cell division of the reproductive cells. It is a process where a single cell divides twice to produce 4 daughter cells. Here the number of chromosomes in the daughter cells gets changed. The number of chromosomes in the daughter cell becomes double to that of the parent cell. Meiosis occurs in sperms and ova. And now this is important, meiosis occurs in 2 steps. Step 1 is meiosis 1, and step 2 is meiosis 2. Let's look in the detail. Okay, first we will look at meiosis 1. This is the first step of meiosis. Meiosis 1 occurs in the following steps. Prophase 1, then metaphase 1, following that is the anaphase 1, and finally the telophase 1. Time to look into the detail about each of these steps. You might have some problem if your basic genetics is not clear enough. Well, as per the request of my viewers, I will soon create a video on basic genetics, so till that time use Google to know a little bit about basic genetics. Prophase 1. Prophase 1 is longer than the prophase in mitosis, and is divided into 5 steps. Leptotene, then zygotene, then pachytene, then diplotene, and finally diakinesis. In leptotene, the chromosomes begin to condense, and attain a compact structure. In zygotene, the pairing of homologous chromosomes starts a process known as chromosomal synapsis, accompanied by the formation of a complex structure called synaptonemal complex. A pair of synapsed homologous chromosome forms a complex known as bivalent or tetrad. In pachytene, crossing over of non-sister chromatids of homologous chromosomes occurs at the recombination nodules. The chromosomes remain linked at the sites of crossing over. Well, you got any doubt? Study about basic genetics first, and then come back here. Continuing. In diplotene, dissolution of the synaptonemal complex and separation of the homologous chromosomes of the bivalents except at the sites of crossover. The X-shaped structures formed during separation are known as chiasmata. Lastly, diakinesis is marked by the termination of chiasmata and assembly of the meiotic spindle to separate the homologous chromosomes. The nucleolus disappears and the nuclear envelope breaks down. Here's a diagram of prophase 1. As I told, the chromosomes condenses, and the nuclear membrane breaks down. Metaphase 1. Here the bivalents align at the equatorial plate, and microtubules from the opposite poles attach to the pairs of homologous chromosomes. Okay, bivalence means the chromosomes. Diagram of metaphase 1. See how the chromosomes have aligned themselves in the middle of the cell. Oh, you saw the crossing over, didn't you? Next is anaphase 1. The bivalents align at the equatorial plate and microtubules from the opposite poles attach to the pairs of homologous chromosomes. Diagram of anaphase 1. See how the chromosomes move to the opposite poles of the cell to get split up. Telophase 1. Well, this is the last step of meiosis 1. the nuclear membrane reappears, and is followed by cytokinesis. This gives rise to a dyad of cells. Here's the diagram of telophase 1. The chromosomes gather at the poles of the cell, and in a single cytoplasm there are two daughter cell structures. Cytokinesis. Well, from the previous video you know what is cytokinesis, and here two daughter cells are formed. But hey, wait, the process of meiosis isn't complete. I know you are quite curious to know how the process of meiosis is completed. But before that, let us quickly recap the process of meiosis 1. It starts with prophase 1, then it is followed by metaphase 1, then anaphase 1, and finally the telophase 1. Hope you enjoyed the journey till here. Now let's enter the world of meiosis 2. This is the second and the last step of meiosis. Meiosis 2 occurs in the following steps: prophase 2, metaphase 2, anaphase 2, and finally the telophase 2. These steps are in order, and so occurs one after the other. Let's look into the detail of each of these steps. Prophase 2. Well, this is same as that of the prophase 1 in meiosis 1, but here this process happens with those two daughter cells, which were formed during meiosis 1. Here the chromosomes begin to condense, accompanied by the dissolution of the nuclear membrane, and the disappearance of the Golgi apparatus and ER complex. Here's the two daughter cells from meiosis 1. Well, prophase 2 has started in each of these cells, and new spindle fibers are formed around the chromosomes. Next, metaphase 2. The chromosomes are connected to the centriole poles, at the kinetochores of sister chromatids through the microtubules. They also get aligned at the equator, to form the metaphase plate. Here's the diagram of the metaphase 2. See how the chromosomes of both the daughter cells has arranged themselves at the middle of the cells. Anaphase 2. In this phase of meiosis 2, there is a simultaneous splitting of the centromere of each chromosome, and the sister chromatids are pulled away towards the opposite poles. As the chromatids move towards the poles, the kinetochore is at the leading edge with the chromosomal arms trailing. This thing will become clearer when you see this diagram of anaphase 2. See how the chromosomes got divided, and are moving towards the poles of the cells. Telophase 2, well, this is the last step of meiosis 2. The chromosomes dissolve again into an undifferentiated lump, and a nuclear envelope develops around it. Followed by cytokinesis, telophase 2 marks the end of meiosis 2. 4 haploid daughter cells are formed as a result. See this diagram of telophase 2. Each of the cytoplasms of the cells got 2 cellular structures each. Okay, this is the final cytokinesis of meiosis 2, and finally meiosis. See how 4 daughter cells are formed at the end of the process. So the formation of 4 daughter cells marks the end of meiosis. This is the entire process of meiosis 2. It starts with prophase 2, followed by metaphase 2, then anaphase 2, and finally the telophase 2. Oh yes, 4 daughter cells are formed during cytokinesis, at the end of the process. This is the entire process of meiosis. See how it starts with just a single daughter cell in prophase 1, followed by metaphase 1, then anaphase 1, and finally the telophase 1. After telophase 1, cytokinesis takes place, and gives rise to 2 daughter cells. These 2 daughter cells are used in prophase 2, then metaphase 2, then anaphase 2, finally the telophase 2. After telophase 2, again cytokinesis takes place, gives rise to 4 daughter cells, which marks the end of the process of meiosis. Let's look at the significance of meiosis. Reproduction in animals takes place through the fusion of gametes, i.e. two cells fuse together with their genetic material, to develop a zygote. Unlike mitosis, meiosis produces 4 daughter cells. Without meiosis, there would be no birth or reproduction. In meiosis 1, the homologous chromosomes separate from each other, whereas in meiosis 2, the sister chromatids separate. Crossing over is the process of meiosis, in which 2 chromosomes of a homologous pair are exchanged between non-sister chromatids. Again an endnote for you. So that's it. Thanks a million for watching this video, and doing a wonder for our beautiful family. I will again meet you in the next video. Till then, just subscribe to our channel and click the bell icon to never miss the upcoming videos from 1234 Media Bio.